Welcome, everyone. On behalf of Cambridge Healthtech Institute's Global Web Symposia series and our sponsor for today, Cellecta Incorporated, I'd like to welcome you to Loss-of-Function Genetic Screens with RNAi and CRISPR libraries. My name is Elizabeth Lamb, and I'm the host and the moderator for today's event. Now I'd like to introduce Kareem Hyder, and I'm going to pass the presenter ball over to you, Kareem. Kareem is the Director of Operations for Cellecta. Welcome, Kareem.
Thank you, Elizabeth. Thank you very much for calling in, everyone, and welcome to our webinar. Today we will present loss-of-function genetic screens with RNAi and CRISPR libraries. As Elizabeth mentioned, my name is Kareem Hyder. I'm the Director of Operations at Cellecta. And before we begin the technical portion of the presentation, I'd like to take a few minutes to give you a bit of an overview of our company, Cellecta. So Cellecta is a small company located in Mountain View, California, about a half-hour south of San Francisco. Our company was founded about 8 years ago by a group of scientists strongly interested in developing more powerful genetic screen tools to advance drug target discovery. Seed funding from the organization was obtained through NIH SBIR grants, and this grant program continues to be an important component of our R&D funding. We remain strongly focused on R&D efforts, with most of our employees actively working in the lab. Our earliest and most developed platform is RNAi-based and more recently CRISPR-based loss-of-function genetic screening, which we'll focus on today. While we continue to push the limits of this technology, however, we're also in the process of developing other complementary approaches to genetic screening and analysis. To augment our R&D efforts, we maintain a diverse network of collaborators, a few of which are listed here. As you can see, many of these groups are focused on oncology research. Disease research in this area in particular makes use of molecular techniques to identify essential genes that could be potential drug targets. Loss-of-function screening is a powerful tool for these studies, but it certainly could be applied to other fields besides oncology. And here's a list of our products and services, just a brief overview. To provide more flexible access to the technology for these types of genetic screens, we have commercialized a range of products and services also. That includes full-service screens, custom libraries, individual constructs and cell lines, and we go as far as complex barcode cell truck libraries. And as always, we'd be happy to discuss any projects you're interested in pursuing, where we think these types of, or where you think these types of screens will be useful in your studies. And now, to give you a better understanding of what loss-of-function screening can offer you and how it can be used, I'd like to introduce our speaker for today. So here's Paul Deal, our Director of Business Development. He'll present our RNA and CRISPR-based loss-of-function screening platform. Paul.
Thank you, Kareem. Thank you, everybody, for taking time out to log in and listen to our presentation of the loss-of-function screening platform, as Kareem mentioned. I'll take you through what Cellecta has developed in this area in terms of this type of screening approach to identify genes that are responsible for various biological processes and responses and can be used to identify novel drug targets. First, let me explain a little bit about how we develop the loss-of-function screening libraries, and that's sort of the core of the platform. Loss-of-function screening is a approach that can be used to identify genes that are responsible for various biological processes by disrupting or interrupting them in cell systems. And this is particularly useful for groups that are interested in identifying targets that are driving, gene targets that are driving disease processes in particular, but any sort of biological or gene disease associations. And so, as Kareem mentioned, a number of the groups that are doing these types of screens are in oncology research. But in addition, there are other areas such as infectious diseases or immunology and inflammation where they are interested in identifying genes that are driving various processes. And so loss-of-function screening then is an approach of disrupting genetic elements and then identifying the ones that are important functionally for the process. This is different than, say, transcriptome analysis, where you're looking at the correlation of genes that are activated or deactivated and correlating that with various phenotypes. For large-scale loss-of-function screening looking across the whole genome, there are really only two technologies that provide a platform to be able to do this. One is RNAi and the other one is CRISPR. And RNAi, as many of you are probably familiar, has been a approach that's been used for a number of years. CRISPR is a more recent approach, but both of these rely on small RNA effector molecules that can be translated or transcribed in the cell and then used to target whatever gene is of interest. And so the ability here is that with the RNAi or CRISPR technology, you can screen then large numbers of genes by simply designing them in silico, designing the effector molecules in silico, and then synthesizing them and introducing them into cells. I'll talk a little bit about the RNAi platform that we've developed and have been working with for a number of years, and then how we're also now bringing the CRISPR platform online for these types of screens, and the comparison of the two. There are two approaches for this sort of screen. One is a RAID format, and in this case, each one of the effector molecules that you're using to target various genes can be introduced to the cells individually in different wells in a large 96-well plate, or larger plates. And so in this case, each one or a few molecules are introduced to the cell separately, and these types of screens then on any sort of large scale, when you're looking at thousands of genes, require a significant amount of infrastructure. It takes a lot of planning, and in terms of flexibility, the screens are somewhat fixed. And so the scalability of this approach is somewhat limited, and you also get well-to-well variation and some differences between the cells, and you have to worry about replicates, etc. With the pooled format approach, it's a little different. In this case, cells are incubated all together in culture, and the effector molecules are all introduced out to the cell population as a whole. So we introduce, you can introduce several thousand different effectors, several thousand different shRNA or sgRNA to the cells, and then look to identify which ones in that large population that's been introduced into the cells are having an effect on the phenotype that you're interested in. And those are the ones then that are important, presumably, for the response that you're looking for. And so the approach is a little different. And the advantage here is it's very flexible because you're using normal cell culture techniques. It doesn't require a large-scale infrastructure. And you can look at your cells one day and you can decide whether you want to harvest them or if they need to continue growing for some reason. And you can then screen a larger number of different cell populations and look for consistency across the populations. In addition, we take the pooled approach for the format all the way back to when we synthesize the libraries, which, as I said, are sort of the central key element in this pooled approach, because we need to have then a large library and be able to introduce that into a large population of cells to do this sort of pooled screening on a genomic scale. And so we start with a pooled approach synthesizing the oligonucleotides. These are done on an array synthesis, and we clone then a large pool of tens of thousands of oligonucleotides at a time into a vector system to make the libraries. And the nucleotides, the oligonucleotides, code for shRNA or sgRNA, or they can code for other effectors too. This type of approach is generally applicable. But in this particular application with loss-of-function screening, the oligonucleotide codes for the shRNA or sgRNA. In the case of the shRNA, we often include a barcode. But then the insert is cloned into a vector, a lentiviral system, and we use a lentiviral system to ensure that we're going to be able to introduce the large number of effectors into a large population of cells effectively. And these are cloned all together, and then the whole library is packaged. And so you may have 50,000 different sgRNA targeting 6,000, 7,000 genes, or you may have, even 100,000 different shRNA targeting particular set of genes. It really depends on what you want to do in your screening. But the advantage here is that we can make this library from start to finish within just a couple of months. And so we can define whatever set of shRNA or sgRNA. We can vary the structures. If there's some particular features we're looking at, we can design all of this in silico, make the oligonucleotides, and then clone them into the vector. And we have a lot of flexibility then in the vector too. And So in our standard vector shown here, we normally have the U6 driving the shRNA or sgRNA, and then we have a reporter RFP, and this is useful for identifying which cells have picked up the virus and are expressing then the effector. And then we also have a selection marker, puromycin. For some screens, it's important then to select the cells that have also picked up the virus and get rid of the cells that are not part of the assay. But we can change this out and actually substitute other markers. We can use different types of vectors, etc. And once the library is made, then we package this library into lentiviral particles. And these are highly efficient in infecting large populations of cells. In addition to the standard vector, we also do have a TET-inducible vector that's shown here. We use this for some specialized screens. Again, this is the advantage of the approach. We can actually make a library in both vectors very easily by cloning the same set of oligos in either one. And then we have, an array of different choices in terms of the vectors that are usable for this. Once the library is made, it's important to make sure that the quality of the library is good. And the first step is, of course, to pick individual clones from the library and sequence them and see what the mutation rate is, etc. And we target a mutation rate that's less than 0.05%, and then 0.5%. And then in addition to that, the library is qualified by next-generation sequencing. And this is a way of doing reads of all of the inserts that are in the library and looking to identify which ones are, what's the frequency of each one of the shRNA species in the library. And this provides a baseline for what the representation is of the library once it's made, and this will also usually be the baseline then for most screens, because then we're looking for, with a selection process or after the library is introduced into the cells, we're looking for changes in specific shRNA representation in the cells relative to the baseline of the library to see which ones are affecting the process that we're looking at during the screen, as I'll show you. But then also this is to ensure that the library is good quality. As you can see here, this is a relatively narrow peak, so there's a narrow distribution of representation for each one of the inserts that are in the library. You can see here a poor quality library that we get a distribution over about 3 orders of magnitude. So this library would be more difficult then to identify enrichment or depletion in the shRNA populations after a screen. And so this library then wouldn't really meet our quality standards. So using this approach, we can make, as I mentioned, within a few months, custom libraries. And then, in addition, we have also made some pre-made libraries. We have shRNA library that covers all protein coding sequences in the genome. That's divided into 3 modules. Each one targets approximately 6,300 genes. It's a 6,500 here. We are also in the process of releasing a CRISPR genome-wide library that is targeting the same set of genes with 3 modules also. So that's the core of the approach, is actually making these quality libraries so that this type of pooled screening is possible. So then how is the pooled screening done? Once the library is made, we have the library packaged here, and as I mentioned, it's cloned into a lentiviral system and packaged in lentiviral particles to ensure that we get high efficiency transduction or infection of cells with the library in the screen. And so once the packaged library is made as lentiviral particles, it's introduced into the cells. And this is a critical step in the process because it's important to have the full library completely represented in the cell population at the beginning of the screen in high enough numbers so that you're going to get robust results at the end. And so we normally recommend about at least 200-fold the complexity of the library. So if you have a library with 50,000 constructs, it's important to transduce at least 10 million cells with that library in order to make sure that you have enough of each one of the sgRNA or shRNA that's in the library in in enough cells so that you can look to see whether you're getting an effect and whether you're getting an effect at the end of the screen, because you're looking for a change in the frequency of each one of the shRNA or sgRNA that are in the library. So after the library is transduced into the cells are treated depending on whatever selection or type of screen that you're interested in running. And normally, the most popular screen is really just to allow the cells to grow over a period of time. And in this case, it's to identify essential genes. So if you allow the cells to grow and identify the effectors, the sgRNA or shRNA that are underrepresented in the population after a period of time, these are the ones that are inhibiting or killing the cells in the screen. And so these are the ones then that presumably are doing that by interfering with the genes that are essential for the growth conditions of these cells. But there are other types of screens also. You can treat the cells with a particular factor and kill most of the cells, or try to kill most of the cells, and then identify shRNA or sgRNA that prevent that effect. And those presumably then are targeting genes that are important for the response that you're looking at, the lethal response in that case. Or you can do a screen where you're looking at a fluorescent marker, a fluorescent reporter, and looking for genes that are essential for the activation or the deactivation of that reporter, and do a selected population. In any of these cases with any of the type of screens, after you've done the selection of the cells, then you isolate genomic DNA and then sequence the sgRNA or shRNA that's present in the genomic DNA. So then what you're looking at is the population of cells that have each particular shRNA or sgRNA after the screen. And then this can be compared with the original frequency of each one of those species in the library at the start of the screen. And you're looking for shRNA that either enrich the cells or allow them to grow under certain conditions, as I mentioned, or are depleted or less represented in the population after selection or after growth compared to the original library. The types of screens then are basically a positive and a negative selection. And you can call them rescue screens or survival screens. And in this case, the screen is done in such a way as to eliminate most of the cells. Or, as it's shown in the third example there, it can be done on a fax, say, with a reporter. And in that case, what you're doing is selecting cells with activation of a reporter. But what you're looking for are the enrichment of particular species of SH or sgRNA in the population compared to either the original population of the library or a control population. The reverse of that is a negative screen where you're looking for essential genes. And in this case, under certain conditions, for example, you might be interested in identifying genes that are essential for growth in the presence of a drug. Or you might just be looking for ones that are essential for growth under normal media conditions. And in this case, what you're looking for is depletion or lower representation of a particular shRNA in the population after the screen compared to the original library, because that indicates that these shRNA or sgRNA are negatively affecting the growth of the cell. So that's a negative selection. And I'll show you an example of that. This is the most popular type of screen, actually, because as Kareem mentioned in the beginning, a lot of oncology researchers in particular are interested in this type of approach to identify vulnerabilities in cancer cells. And so in that case, what they're looking for are genetic targets that, when they're disrupted, interfere with growth of the cell or kill the cells. And so this is a screen of K562 cells, which is a chronic myelogenous leukemia cell line. This is the data from the screen, and you can see each one of these small dots on the graph here represent the counts, the number of reads from next-generation sequencing for a particular, this is an shRNA screen, shRNA, a particular shRNA in the population after a period of growth, either 2 days of growth or 9 days of growth, versus the control, which is essentially the baseline or the original frequency of that particular shRNA in the library. So you can see that most of the shRNA are on the diagonal. That indicates that there's really been no change from their representation in the original library versus the representation in the population after growth. And that's what you would expect, because most of them then are not interfering with growth of the cells. The darker spots on this graph here are showing the representation or the counts for a control that's contained in the library. These are various shRNA that are targeting the luciferase gene, which is not present in these cells. So this gives a sense of what the background level is in the cell. And so then you can see that there are also a number of dots that are highlighted in blue on these particular arrays, these particular graphs, that are appearing below the diagonal. And so that's indicating that these are underrepresented in the experimental population versus the control, which means that they're interfering with growth of the cell. And presumably they're doing that then by interfering with the target gene, the gene that they're targeting, and that gene is essential for the growth of the cell under these conditions. So this is often called a dropout assay, a viability dropout assay. And you can see over here on the right that there are, with this particular library, this is a library that has 27,000 different constructs in it. There are 5 to 6 of them targeting each one of the genes, and it's targeting approximately 5,000 genes. And you can see that after 2 days of growth, there are some of them that are targeted. Some of the shRNA targeting the same gene are showing up in the hits. And you can see after about 9 days of growth, we're actually getting quite a few where there are multiple shRNAs targeting the same gene that are showing up as hits in the screen. So we took a look at these ones where, and that's what you want, because if you get a single shRNA that's giving you a dropout, it presumably, hopefully that's happening because it's targeting the gene, but also there may be some off-target effect or some other effect that's spurious. So it's useful to have multiple effectors targeting the same gene, and then if you're seeing those show up in the selected population as important, then you can be more confident that it's actually the target gene that they're interfering with that's actually giving you the effect. And so we took a look then at the positives in this screen, the ones especially that had 2 or more shRNA that indicate that those genes are important. Oh, and I just wanted to point out that these assays are typically done in replicate. In this case, this is 2 replicates, and you can see that the results are very robust, and that we're getting a correlation of better than 0.9. And so the assay itself is actually giving consistent results. And that's because we're getting good representation of the library in the cells at the start of the assay. When we took a look at the hits from the K562 cell line, we did find that a number of them had annotations that were associated with leukemia or other blood cancers. We identified BCR and ABL as essential genes, and there is a BCR-ABL fusion rearrangement in this cell line. That's an essential driver for K562. These hits then did seem to make a lot of sense with the screen. And we did this sort of screen with several different blood cancer cell lines. We were looking at actually trying to identify genes that are important in blood cancers. But probably the most interesting outcome of this study was we began working with a group at Huntsman Cancer Center, and they had a K562 cell line that was resistant to imatinib, which is a standard of care for chronic myelogenous leukemia. And they did a similar sort of screen with the same library to identify sensitivities in the resistant cell line that could be specifically targeted and maybe as complementary or follow-up to the imatinib therapy. And they were able to identify that the imatinib-resistant cell line is still vulnerable to thymidine kinase pathways and inhibitors. So, I'm sorry, not thymidine kinase. They were able to identify a sensitivity in the cells that became resistant to the thymidine kinase inhibitors. And so that's one example of how this type of approach could be used. Another larger example is a group at Novartis that screened large numbers of cell lines with various different genetic lesions, looking for sensitivities that correlated with the different lesions in the cell lines. And in this particular study shown here in PNAS, they identified cells with the SMARCA4 mutation or lesion are highly dependent or sensitive to disruption of the SMARC2 gene. And so this is a sensitivity that's associated then with this lesion in the cell line and may represent something that could be exploited therapeutically. Another example, another approach that can be used for this sort of screen is to identify genes that help make cells resistant to particular drugs, and when they're knocked out, increase sensitivity to a drug. And this is a screen done by the German Cancer Institute here, where they looked at pancreatic cancer cells treated with a gemcitabine to see if they could identify sensitivities that would make the cells more sensitive to gemcitabine. and they were able to identify that. So these are just a couple of examples of the type of loss of function screens and the results that can be found with them. And there are several other examples. Some of them are shown here on this slide, and then they're also on our website. And so the shRNA screening has been used for a number of years, and it is a proven approach to identify genes that are driving particular responses or important for biological responses, as opposed to, as I mentioned, transcriptome profiling or other techniques that are really looking at correlations and are looking at genes that are activated or deactivated in conjunction with those responses. More recently then, CRISPR technology has been developed, and if you're familiar with this technology, the CRISPR system, which has this long, clustered, regularly interspaced, short palindromic repeat name that's very difficult to remember, so it's just called CRISPR, was originally identified in bacteria quite a few years ago. And it's a defense mechanism that the bacteria use to chop up nucleic acids from invading viruses. However, a few years ago, researchers at the University of California and I believe University of Austria found a way to transport this system, how the system works, and actually be able to take the system out of the bacteria and put it into eukaryotic cells so that you're able to use it to specifically target and disrupt genes at the genetic level. So this is a knockout system. And this system, like shRNA, uses a short RNA sequence with a guide that targets a particular sequence. This is a 20 base pair guide with sgRNA that we're showing here. The difference here between shRNA and the sgRNA system is the shRNA system uses a endogenous pathway that's in mammalian cells already to interfere with the transcript level of the gene and have that degraded. sgRNA, or CRISPR, on the other hand, uses it requires a nuclease to disrupt the gene that's targeted on the genetic level. And so in addition to the shRNA, it requires that the Cas9 nuclease also be introduced into the cells for this targeting disruption for the knockout to work, the disruption of the gene. However, because of the fact that it's using the short the short RNA sequence to target and disrupt the gene, again, large libraries of sgRNAs, guide strands, for the CRISPR system can be made in silico. They can be designed, they can be synthesized easily, and they can be introduced into cells. And so this makes it amenable for the type of loss-of-function screening, similar or analogous to what we've been doing with shRNA for a few years. And at the beginning of last year, 2 groups in Boston actually published genetic screens using the CRISPR-Cas9 system. And so this, of course, piqued our interest because this is offers a orthogonal approach to do the same type of loss-of-function screening. And so based on these publications, we moved forward with developing a Cas9 CRISPR system so that we could try this similar sorts of screens. We made a single vector construct with the a Cas9 and the sgRNA coding sequence that could be inserted under the U6 promoter like it is with the shRNA. And then we also have a puromycin selection in this construct. And we, after developing this, introduced various sgRNA to a GFP reporter, and you can see here in the first panel cells that have the GFP are expressing the GFP integrated into the genome. This is a stable cell line. And then when the CRISPR construct expressing SG targeting that GFP is introduced into the cells, you can see we're getting pretty complete knockout after 7 days in the cell culture. Notice it's not 100% knockout. Some of the cells are still expressing GFP, and this is what you would expect. So I just want to make a distinction here. One of the important differences between CRISPR and shRNA is that with CRISPR, in each particular cell, you're either getting a full knockout, it's a knockout, no expression of the gene, or no expression of that protein is no longer existent because it's knocking out the genetic element that's making the transcript for that protein. But if you go across the population, if you introduce it into 100 cells, not all 100 cells are going to get the knockout. There's going to be some small percentage of the cells where the knockout doesn't take effect, or possibly you get an in-frame mutation, and so you're still getting expression of a transcript that's pretty much the same protein. This is different than with shRNA, where you're in a cellular level, you're not getting complete interference or elimination of the transcript, but you're getting most of the transcript interfered with. And so when we're talking about 90% or 95% of knockout in a CRISPR system, what we're talking about is across the cellular population. Each one of those cells is either on or off, but we're getting across the population about 90% knockout. And that's what's shown in this example here. More quantitatively in the next slide, you can see that this is the percent of GFP knockout cells, and you can see that after 9 days we're getting well over 70, 80% in most cases knockout, where after 14 days we're getting over 90% knockout in the cell population. So the longer the cells incubate with the sgRNA and Cas9, the more of the genes that are being knocked out. And these efficiencies are critically important if you're going to do a loss-of-function screening, because you need to get a high percentage of knockout across the population in order to be able to pick up the signal, to pick up the effectors that are having an effect on the cells in this type of assay. We also made a version of the single vector construct with the UBI promoter. This is the ubiquitin C promoter, which is not as strong as the CMV promoter, especially in these cells. These are 293 cells. And you can see that we're getting different levels of knockout depending on the amount of Cas9 that's expressed. This is the promoter that's driving the Cas9. And so that suggests the Cas9 expression is the rate-limiting step in terms of the knockout across this population. One of the issues, though, with the lentiviral system, and it is critically important to do a lentiviral system for this type of screen, as I mentioned, because we need to introduce a large heterogeneous population into a large number of cells. One of the issues with the lentiviral system is that even though it can take a relatively large payload of nucleic acid, there is a limit to how much can be packaged into the lentivirus. And so the Cas9 protein itself is quite large. It's about 4.5 kb. And then in addition to having a selection marker and then some other genetic elements in there to ensure expression and packaging, proper packaging, we're approaching the limit that can be effectively packaged in a lentiviral system. And so for this reason, thinking that we're interested in screening large numbers of cells, and we need to make possibly 10 million viral particles per infection in a screen, we were getting low titers with this single vector construct. And so we moved to a 2-vector construct for screening purposes. And in that case, what we did is we separated the Cas9 and put that on its own construct with a hygromycin marker. And then we have the sgRNA construct. And this is very similar to our shRNA expression vector. And in this case, we have now the pure myosin selection, but we also now have room again for the fluorescent protein so that we can readily identify cells that have been transduced with the sgRNA. Then the way this screening would work is the Cas9 is transduced into the cells initially, and once the cells are selected for Cas9 and they're expressing the Cas9 protein, then the sgRNA can be introduced to do the actual screening, the genetic screening. So after making the 2-vector system, we tested it and we found that we are getting high titers with the 2-vector, with the sgRNA vector. These are similar titers as we get with the shRNA vector. And then the Cas9, of course, gives lower titers, but that's not really a problem because in this case we can introduce it into a smaller population of cells and then we can do a selection. So we can ensure that all the cells are expressing Cas9 before we move forward with the screen in the sgRNA vector. The 2-vector system gives significant knockout. Here you can see the 9 days and 12 days again in a GFP system. So this is the level of fluorescence of knockout of GFP in that system for those 2 different days. And so we're getting effective knockout with sgRNA in the 2-vector system. And in fact, since we have more room now, we can also use the inducible approach that we used with the shRNA and make an inducible sgRNA. And that also works to give inducible knockout if that's necessary for certain types of screens. And so we have actually both of those options available. But when we look at knockout with the two-vector system, one thing we did notice is that we're getting somewhat stronger knockout with that system than we're getting with a single-vector system. And on reflection, we realized that this is likely because we have the Cas9 being transduced into the cells, and we can transduce that at a somewhat higher level, a higher MOI, than we can transduce the library itself. And we can also select for Cas9 more strongly and make sure that the cells are expressing it. And since there's some indication from the previous data that I showed you that the Cas9 level affects the rate of knockout of the cells, we then thought, well, this is probably, since we're getting better expression of Cas9, we're getting stronger knockout across. And so this would be better for a library screen. And we took a look at that by transducing cells with high numbers of Cas9 versus a single copy of the Cas9, or an MOI of three copies of Cas9 per cell. And you can see that when we increase the level of the Cas9 gene that we introduce into the cells, we're getting more effective knockout more quickly across the cell population. So although we made the change to a two-vector system for sort of practical reasons in terms of packaging, it also seems like this may be a preferable way of doing an sgRNA screen because you're going to get quicker knockout more rapidly. So with that, we went on then to we went on to compare the shRNA library screen with the CRISPR library screen. And I had mentioned previously that we have a CRISPR genome-wide, an shRNA genome-wide library targeting all the protein coding genes divided into 3 modules. So we took the first module that targets approximately 6,300 genes with 8 shRNA each, and we made an analogous version of the sgRNA library. So we're targeting the same set of human genes in this case, and we have 8 sgRNA targeting each one of those. And using this library, then, we ran the screen, and to do this, we used actually CML cells. These are actually patient-derived CML cells that have been passaged through mouse. And so they're similar to the K562 cells, but it's not a cell. It's a cell line that was generated from tumors from an actual patient. And we actually have 2 versions of this. One is resistant to the treatment, the imatinib, and the other one is sensitive to the imatinib. So there's actually 2 sets of screens here we're looking at. The cells were transduced with Cas9 initially for the sgRNA screen at an MOI of 2. This is multiplicity of infection. And then they were grown up, and both populations of cells were transduced with shRNA and then separately with sgRNA to do separate screens, a CRISPR screen and an RNAi screen. And the transductions were done at 400 cells per construct. And then after selection, they were grown and harvested at 3 different time points, 2 weeks, 3 weeks, and 4 weeks, to take a look at what sort of hits that we're getting, what sort of depletions are showing up at these various time points. And this is just some representative data of the screen. You can see the shRNA screen on the left and the Cas9 screen on the right. The red dots are showing the positive controls, so we're getting dropouts of genes that we know should drop out in the screen, and that's indicating that, holistically at least, it seems to be working. When we looked at the controls more quantitatively, what's shown here are plotted are the negative controls and the positive controls that we have in the library for each one of these SG or shRNA libraries for each one of the screens. And we found that actually we're getting good response in both cases with the shRNA and the sgRNA. With the shRNA, you can see that it actually almost looks like you're getting a slightly stronger response in terms of a negative fluoride depletion at the log 2 here than you're getting with the shRNA. But the negative controls didn't really give any response with either screen. And so the screen itself seems to be working. What's interesting is that the error bars with the shRNA seem to be much broader than they are with the sgRNA. And if you look at the next slide, it's shown a little bit more quantitatively here, where we're looking at the mean difference between the negative and the positive controls at the various time points. You can see that at the 2-week time point, the results are pretty close. But at the 3-week time point, for example, the shRNA statistically significant depletion that's measurable is less than it is with the sgRNA. And that's because of the error bars. What that's showing is that we're getting more noise in the assay with the shRNA at the 3-week and the 4-week time point. And this doesn't seem to be occurring with the sgRNA. And so this suggests that the sgRNA can go out for a longer period of time, and the assay itself remains less noisy. It also suggests that you want to probably go a little bit longer with an sgRNA than you do with an shRNA screen, because we're getting a stronger signal at 3 weeks than we're getting at 2 weeks. And this is consistent with the results that I showed you in terms of the knockout. It takes about a week or a little over a week to get a strong knockout across the population with sgRNA with the CRISPR. Where with shRNA, we know from experience that within a couple of days you're getting the maximum knockdown that you can get. So then the question is, the controls seem to be working, and both screens seem to look pretty good. The question is, what type of hits do you get out of both? So we picked the 2-week time point for the shRNA and the 3-week time point for the sgRNA screen, because that seemed to be where we were getting the best differentiation between the controls with both assays with the least amount of noise, and analyze the hits that we're getting. And if we just look at the hits of the constructs in both of these assays, you see that the shRNA is actually on an shRNA level, on a construct level, effector level, is giving a much higher number of hits. We're getting about 10% of all of the constructs that are in the assay are showing depletion, are showing a significant reduction in their level relative to the control, or relative to the mean. And so, whereas we're getting much less of this with sgRNA, although, so it seems like we're getting about 3% or 2 to 3% of the sgRNA are giving significant hits. And this is suggestive of a fair amount of noise in the assay, which is known for shRNA, because if you take a look at the shRNA or the sgRNA that are targeting the same gene, and which ones are giving hits, which genes are getting hits from multiple shRNA or sgRNA, you can see that on this graph here, that only about less than, say, half of the library of shRNA is not giving any signal whatsoever. And about half of the sgRNA, about 40%, are actually giving some significant depletion over the time course here, compared to just a small percentage of the shRNA. But these are single hits. So for this particular gene, it's only one shRNA or sgRNA is actually giving a signal, and the other 7 are not. And so that indicates that, that possibly that this signal is not coming from knockdown or interference with the target, but it's actually coming from, maybe some other off-target effect that particular effector is having on the cell. And if you go up, there's a smaller number, but still a significant number, that more of shRNA where you're getting 2 hits per gene, targeting 2 shRNA that are targeting the same gene. But the story changes a little bit when you go up into the higher numbers. When you're looking at 3, 4, 5, up to 8, multiple sgRNA or shRNA that are targeting the same gene that are giving a significant signal in the screen, we find that the numbers are actually not that different between the shRNA and the sgRNA. The CRISPR screen and the shRNA are both giving about the same number of hits. And so that's just in terms of numbers. The question is then, if we look at the actual targets that are identified in these screens, what's the overlap? And so when we're looking at this and we looked at relatively strong hits here, when we're looking at these strong hits, we find that there are just under 100 genes that seem to be essential in the cells with the shRNA and the sgRNA screens. And if we look to see which ones actually overlap between the two, we're getting about half of them seem to overlap, about 50% in both screens. And this is important because this is showing that both screens are showing relatively the same set of hits, or the same set of essential genes. And the RNAi, as I mentioned, has been used for a number of years and is sort of proven. And then the CRISPR is a newer technique, but there seems to be some interesting features of it. So we're getting 2 different approaches that are showing a similar set of targets, a similar set of essential genes. And this is a relatively strong selection. So if we, for example, look at the shRNA hits and then relax the criteria slightly on the sgRNA side, you're actually getting, you can actually identify significantly more. So we're getting a large majority that are showing up in both screens. by the same token, if you take the sgRNA and relax a little bit the criteria for the shRNA hits, you can start to see that a number of the ones are just below the threshold that are showing up in the sgRNA screen. So really a majority of the hits are actually showing up on both screens. The essential question is, or maybe the interesting question is, are there hits that are showing up in one screen that are not showing up in the other screen? And to do that sort of analysis, you don't want to just look at the ones that are very strong, that show up in a strong selection in one hit and just miss the mark in the other hit, in the other screen. That's not really the approach. The approach is actually then to take a look at the ones that show up in one screen, say the shRNA screen, but you can't find in the sgRNA screen, that if you look at the effect, the sgRNA that are targeting that gene, that they really don't give any signal at all. So you have to look at sort of the bottom of the barrel on the sgRNA side with the top of the barrel on the shRNA side, or vice versa. Look at the top of the hits on the sgRNA side and look at the ones that are at the bottom and show no statistically significant response on the shRNA side. And when we do that, we find that there are actually a handful of hits that seem to show up in the CRISPR screen that did not show up in the shRNA screen. When we looked at vice versa, we saw there were very few of the RNAi hits that did not show up in the CRISPR screen. And so these are listed here. You can see for the CRISPR screen, there are about 8 different targets that seem to only show up on that side and didn't really show up significantly on the RNAi side. And a number of these, actually, a couple of them have been identified by some people some collaborators that we work with as chromatin modification enzymes. And these are difficult to knock down with shRNA because even if a small amount of the protein is present, you're still getting enough of an effect to enable the cells to be viable. And I'm sure that some of these other hits in this category are similar, that you can get knockdown of the target, but even with significant knockdown of the target, unless you completely eliminate it, you're not really seeing a lethal effect. On the RNAi side, there was only one that was common, that was the ubiquitin C gene here. And the interesting thing about that is structurally that's transcribed in tandem, so there are actually several copies of the gene on the transcript. And so it may be that trying to knock that out with sgRNA is more difficult because you may be able to knock out one of the copies of the gene on the transcript, but there are actually several there. And so that didn't seem to show up too well in the CRISPR screen. But I think that the main message here is that the overlap between the two is actually pretty significant, and they're both showing that we get pretty good results with them. And they're both showing that they can identify essential genes, and they're showing it in a complementary fashion. So then the hits that you get out of the screens are doing both screens is actually maybe you can have more confidence in that because you have 2 different approaches giving you the same answers. And then I'd like to just show the last slide here with for CRISPR, showing that we have the CRISPR screen, the libraries, we now have the knockout constructs and the cell lines, and so we sort of had a complete offering. And then I just added a couple more slides here at the end that I won't go into here, but this is a PCR, next generation sequencing assays to profile markers in cancer cells. You can take a look at these, and I would be happy to tell you more about these. If you have any questions, just email me. My email is shown here on the last slide. So thank you for your attention, and sorry I ran a little bit over on that, but I'd be happy to try and answer any questions we have in the few minutes that are left.
Thank you very much, Paul. We have quite a few, but I think we can probably get to 2 or 3 of them if all goes well. The first question is, what technology has been shown to be more reliable?
Well, I hope I answered that to some extent with the results of the screen. I was scrolling back, but I'll just. I hope I answered that to some extent with the results of the comparison screen. And at this point, I would say that it's not clear that both approaches are giving answers that seem to be biologically significant. And so it's going to take a little bit more studies to show whether there's an advantage to one over the other for certain types of genes or certain classes of genes, or maybe more readily identifiable, or certain hits are more useful for therapeutic applications, for example, with one screen versus another. But I think at this point, it's interesting to look at both the results of both, and you then have 2 different approaches that are giving you essential genes, and that you can be more confident then in the results of the screens by comparing the results of the two different approaches.
All right. The next question. If the ratio between cells and library complexity is too high, is it possible that one cell can be hit by multiple CRISPR or shRNAs?
Yeah, this is actually the question that comes up a lot. It is. We actually usually do an infection at about 0.5 MOI or 50%, about half the cells are infected. And at that level, you do certainly get some percentage of the cells that have more than one CRISPR or more than one sgRNA. But it's across the population. And so if you have one sgRNA that goes into 100 cells, for example, and 20 of those cells also pick up a second sgRNA, the likelihood that any of those 20 are going to pick up the same second sgRNA is very low. So you're not really going to see confounding sort of effects this way. The problem is if you go really high, if you went, of course, to MOI 3 or 5 or something like this, where you're getting most of your cells with 3 or 5 different constructs, 3 or 5 different sgRNA, then in that case you may have a problem. But when you have a small percentage of cells that are infected with, two different of the effectors, it's not really a problem because the what you're looking at are independent. Does that make sense?
It does.
Okay.
And we have time for just one more question. Could you provide some examples of the CRISPR knockout in patient-derived primary cells?
Well, that was actually the example that I gave.
Okay.
That was these were patient-derived primary cells. They are blood cancer cell line, but it is patient-derived, and they were passed through mouse, and then cell line was derived from that. And as I said, we have the resistant cell line and the sensitive cell line. So.
Okay, perfect.
You can try one more, though.
Okay, one more. How do you design your sgRNA to mitigate the risk of off-target effects?
Yeah. Well, it's difficult to do that because no one is really clear even at this point how the off-target effects with sgRNA occur and what type of off-target effects there are. But with both shRNA and sgRNA in this type of screen, the short answer is that you have multiple sgRNA or shRNA targeting the same gene. And so you're looking in the results for ones where you have multiple hits on that gene. And that's, as I showed you here, where we had at least 4 different sgRNA or 4 different shRNA that are giving us significant depletion. Those are the ones that you consider positive. If you just have one lone sgRNA or shRNA and the rest of the ones targeting that gene don't give you any signal, then it's more than likely that you're getting an off-target effect there.
Perfect. And you brought it to wrap up right on time.
All right.
I'd like to thank you, Paul and Kareem, along with Cellecta, for sponsoring today's web symposium. With that, I'd like to say thank you to all of you for coming and sharing an hour with us. I hope we were able to provide some insights that will assist you in your ongoing challenges. I'd like to say thanks and have a great day.
All right. Thank you, everybody. And thank you, Elizabeth, for helping coordinate all this.
You're very welcome.
Bye.